Let's study about the fluorescence application in bioimaging. Many colorful materials has been utilized to stain biological samples. Here is one typical example for bacterial staining. This staining is called as gram staining. Bacteria can be classified depending on this staining pattern, and if it is very positive, strong staining, this bacteria is called as gram-positive bacteria. And if the staining is very weak, like just a pinky, then this bacteria is gram-negative staining. Interestingly, this gram is the person's name. Dr. Gram invented this reagent, and that's why this is called as gram staining. Even before we understand how this gram staining works, we divided bacteria into two big groups. This becomes the standard method of bacteria classifications. This is a typical example of how technology decides scientific development. Another important point here is, can you see the background of these images? Image background is a white color. That means this image is based on subtractive colors. How about this image? All of a sudden, whole background is dark. This image is a fluorescent image. Because of this dark background, fluorescence images usually have low background problem and high sensitivity. Through the electron microscopy images, we know inside the cell there are many organelles. So there are nucleus and also mitochondria and endoplasmic reticulum and lysosomes. But if you take the images using optical microscope, for example, bright field images, first of all, the resolution is quite poor, but this is a physical limitations depending on the wavelength of the light. About the half length of light wavelength is a physical limit of resolution. Also, the contrast is quite poor, and it's very difficult to see whether which part of the cell is a nucleus or other organelles. By using different tricks, so-called phase contrast to the imaging, it's a little better to show those organelle structures inside the cell. Still, it's quite difficult to see which organelle is which. Let's start with the nucleus. How about these images? Comparing to the bright field images, now the nucleus structure is very clear, and this is possible due to this kind of fluorescence dyes. DePi dyes can stain dead cells, and Hux dyes even can stain both dead cells and live cells. Thanks to these beautiful dyes, the nuclear staining in cell imaging is relatively straightforward. How about this? In this image, the red color shows dead cells and green color shows live cells. The red color dye is a propidium iodide, which can penetrate only broken membrane and then stain the DNAs. And this green dye is a calcanein ester. They can enter into the cell, and then by S-rays, those ester bond is cleaved, and then this green dye can be captured inside the cell. Only when the membrane structure is intact, they stay inside the cell. When the membrane is broken, green dye is poured out, and red dye can come in and then stain those nucleus. And in one shot of images, you can easily tell which cells are dead and which cells are alive. Another case is for nucleolus. Inside the nucleus, there is another smaller structure, so-called nucleolar structure. And this nucleola is the center of ribosomal RNA synthesis. That's why this site has very high contents of RNAs. And utilizing RNA dyes, we can selectively visualize nucleolar structures. In this picture, now we can see nucleus in blue color, and red color is mitochondria, and here the green color shows Golgi structures. This image shows the network structure of endoplasmic reticulum, and rhizosome structure as a dot structure like this, and also plasma membrane structure, nucleus, and mitochondria at the same time. Utilizing this kind of organic selective dyes, it's very easy to show 2 colors or 3 colors of a different organelle at the same time in fluorescence microscope. Not only those structural staining, now also the functional molecules can be selectively stained. Calcium is a very important second messenger inside the cell. That's why their measurement of changes in terms of time and space is quite important questions. Here is a report for one calcium dye, Fluorescent. And this dye work has been published in Journal of Biological Chemistry, which is a quite decent journal, but it is not Nature or Science. But the importance of this work was so big. Practically, we can say this single dye could open up the whole field of calcium-based signaling in biology. And the scientist's name is Roger Chen. I expected he get Nobel Prize for this. Well, he got Nobel Prize, but not for this work. He got Nobel Prize for different work. We are going to see his another important work for the Nobel Prize later part. But now let's enjoy about those calcium signaling power using this Fluor dyes. I showed you two examples of dynamic changes of calcium utilizing these calcium dyes.